All right, thank you for the introduction. I'm going to talk a little bit about some interesting results that we've seen from a recent analysis of previously unreported Stanley Miller experiments that were conducted in 1958. As many of you are well aware of, the classic Urey-Miller experiment, published in the 1953 edition of the journal Science, reported several findings of amino acids, including glycine and alanine, using the spark discharge apparatus shown up here in the corner. However, more recently, there was another set of experimental samples found from the volcanic experiment, and it used a similar apparatus to what was used in Miller's classic experiment, except it incorporated an aspirator down here. And what that did was that effectively injected a jet of steam into the discharge chamber. And this study found, using modern analytical techniques, it found over 5 times as many organic compounds as what was found in the original apparatus experiment, and including certain compounds that had never before been produced from spark discharge experiments designed to simulate the early Earth, including homoserine and ornithine. However, even more recently, we found another set of samples that Miller had created, this time from a 1958 experiment. And from confirmation with his original laboratory notebooks, we found that these experiments and these samples were produced using a version of his original apparatus and a gas mixture that included both oxidized and reduced compounds. These gases were methane, ammonia, CO2, and hydrogen sulfide. This was the first time that Miller had used any sulfurous gases in his simulated early Earth's atmosphere, and this experiment was allowed to carry out for a period of 3 days, and as I'll show momentarily, we found a lot of organic compounds were produced using this simulated mixture. So to go over the methods, we used this very similar methods to what was used by the Johnson et al. Paper in 2008, but briefly, we used a combination of both high-performance liquid chromatography with fluorescence detection and ultra-performance liquid chromatography with fluorescence detection, coupled to a time-of-flight mass spectrometer. The liquid chromatography methods used in this paper used a fluorescent agent known as OPA-NAC, which tags primary amino groups. So secondary amino acids such as proline and sarcosine were not targeted in this study, but the use of both liquid chromatography methods and mass spectrometry methods allows us to obtain both chromatographic and mass spectral identification of these targeted compounds to overcome any coelution interferences that may be present from using just liquid chromatography methods alone. So the results of this study were that we found a wide range and abundance of organic compounds. We found 23 amino acids and 4 amines. 7 of these 27 compounds were sulfur-containing organic compounds, including 1 sulforamine and 6 sulfur amino acids. And as you can see from the figure here on the right, we have chromatogram trace A, which is a composite sample chromatogram trace, and we have trace B, which is a composite amino acid and amine standard, and trace C, which is an analytical reagent blank. It's important to note that we did not find any procedural blanks in the sample set that Miller had saved from his 1958 experiment, so we couldn't run those. But this is a reagent blank to show that the chemical reagents that we were using to analyze these samples contributed very little to the presence of organics inside the samples. There had been other experiments that used a very similar simulated prebiotic conditions to what Miller emulated in this experiment. However, none of them were able to synthesize sulfur amino acids. As a result, this 1958 experiment marks the first synthesis of sulfur amino acids from simulated early Earth conditions in a spark discharge experiment. Here's a plot of the sulfur amino acids and sulforamine that we detected. We found 7 altogether. The 2 most relative abundance were methionine and cysteamine. And methionine is one that's of particular biological importance because it is the product of the start codon that in the genetic code that tells the cells machinery to start translating a design for a protein. Other important sulfur compounds that we looked for but were not able to detect in this study include cysteine, homocysteine, and cysteine. Cysteine and homocysteine we couldn't find directly, but we did find cysteamine, which is a decarboxylation product of cysteine, and we did find homocysteic acid here, which is an oxidation product of homocysteine. This provides some evidence that these samples may have initially been composed of both cysteine and homocysteine. However, due to not being stored in anoxic conditions, it's possible that these products were initially degraded to these other products. So to see sort of where the H2S experiment ranks relative to Miller's other experiments, we did a comparison between the results found in the H2S experiment and those of some of his other discharge experiments, namely the classic experiment, the volcanic experiment, and his silent discharge experiment. And we can see the results of the H2S experiment here in the darkest shade are in large part greater in abundance than what was found in his other experiments. And these marked with asterisks here represent species that are over an order of magnitude more abundant than what was found in the other experiments. Certain species of note that are particularly of greater abundance are certain non-protein amino acids, namely alpha-aminobutyric acid, which is hundreds of times more concentrated in the H2S experiments than in his previous ones. Another interesting note is that this experiment, like the volcanic apparatus experiment, was able to synthesize certain amino acids that had not been detected before from spark discharge experiments designed to simulate the early Earth, and the ones that this one synthesized are threonine, leucine, and isoleucine. In 1972, Miller and some of his co-workers found that if you passed reduced gases by a spark discharge, you can generate amino acids in relatively similar abundances to what was found in a Murchison carbonaceous chondrite. And so to further examine this surmise, we've made a comparison between what was synthesized in the H2S experiment and what has been found in not only the carbonaceous chondrite Murchison, but also in other carbonaceous chondrites as well. And what we found is that the H2S results here in the darkest shade are very similar in relative abundance in terms of amino acids that were detected similarly amongst all samples. They're very similar in their relative abundance to what's found in carbonaceous chondrites. And what this may suggest is that H2S may have some sort of role in enhancing the synthesis of certain amino acids in carbonaceous chondrites and also in the experiment itself, those being the alpha, beta, and gamma isomers of aminobutyric acid, the alpha isomer of amino isobutyric acid, alanine, and beta-alanine as well. So some of the big picture implications of what this study may hold in terms of prebiotic molecules on the early Earth is that although a reducing gas mixture is not believed to be common on a global scale on the early Earth, it may have been common on a more localized scale, such as near volcanic systems or island arc-type systems that commonly emit reduced gases, possibly similar to the mixture that Miller used in this study, including methane, CO2, and hydrogen sulfide. And so what this study can tell us is that although we don't know what the chemistry of primitive volcanic plumes was composed of, it may have been composed of a mixture similar to the one that Miller used, and if so, the results from this study would suggest that these types of chemistries that take place in early volcanic systems may have played a role in producing not only a large abundance, but also a large array of various prebiotic molecules on the early Earth. And as we see from comparison with various carbonaceous chondrites, the similarity between the carbonaceous chondrites and the H2S experiment is that sulfur has been found in carbonaceous chondrites and also in the solar nebula as well. And the similarities between the relative amino acid yields between the carbonaceous chondrites and the H2S experiment suggests that H2S may have played a role either on the parent body of the meteorite itself or in the solar nebula in enhancing the synthesis of certain amino acids. So with that, I'd like to acknowledge some people that have been very helpful throughout the process of this study and getting this work completed. I'd like to thank Dr. Jamie L. Sila at NASA Goddard. I'd like to thank Nick Hudd and Facundo Fernandez at the Georgia Institute of Technology. I'd also like to thank funding sources, NSF and the Center for Chemical Evolution at Georgia Institute of Technology, and the NASA Astrobiology Institute. I'd like to thank Mandeville Special Collections Library at the University of California, San Diego campus for archiving and making accessible Miller's original laboratory notebooks. And of course, without Miller's diligence and his dedication to his work, none of this new information would be possible. So with that, I'd like to thank you for your attention, and I'll be happy to take any questions.